To begin with, the rules for designing any kind of survey are the same. The survey may be for research in any field. For example, a nutrition survey or a diet survey or a public opinion poll, customer satisfaction survey, for program evaluation like government, healthcare, education or non-profit agency. The steps in designing all surveys are the same. There are 7 steps which, if followed carefully, will lead to a good piece of research work. These 7 steps are: first, define the goals of research. Next, convert the goals to research questions. Then, convert the research questions to hypotheses. Fourth, convert the testable research questions to null hypothesis. Fifth, write the individual survey items, that is the questions to answer the research question. Sixth, test your survey until you are satisfied with its validity. And finally, test the reliability of the survey. The first 5 steps are explained in other modules. This lesson pertains to the last 2 steps relating to the validity and reliability of the survey. The impact of the results of a research project or interpreting the findings is dependent upon these 2 concepts: validity and reliability. Validity. Essentially, validity entails the question: does your measurement process measure what you intend to measure? The related topic: does your measurement process, assessment or project actually measure what you intend it to measure? The related topic of reliability addresses whether repeated measurements or assessments provide a consistent result given the same initial circumstances. In research, validity has 2 essential parts: internal and external validity. Internal validity encompasses whether the results of the study, example, the mean difference between treatment and control groups, are legitimate because of the way the groups were selected, data was recorded or analysis performed. For example, a study may have poor internal validity if testing was not performed the same way in treatment and control groups, or if confounding variables were not accounted for in the study design or analysis. External validity, often called generalizability, involves whether the results given by the study are transferable to other groups, that is, the population of interest. A study performed exclusively in a particular gender, racial or geographic subgroup, such as females in India, may not be applicable to Hispanic men in the Northwest. It is through proper study design and strict protocol execution that high levels of validity, both internal and external, can be achieved. An important point to remember when discussing validity is that without internal validity, you cannot have external validity. Results of a poorly designed or executed study are not applicable to any population in that particular study sample or otherwise. A common threat to internal validity is reliability. Assuming the same initial conditions for a test assessment or process, the test must provide the same results every time it is performed for it to be deemed reliable. For example, if a research protocol dictates that subjects must have their weight measured, the scale should provide the same weight if repeated measures were taken at the same time. Reliability. Reliability is often at risk when assessments are taken over time, performed by different people, or the assessments are highly subjective. In the weight example, if the subject had their weight taken monthly over a period of a year, the scale may lose calibration over that time, or differing clinic staff may read the scale differently over the study duration. Often, physical assessment techniques such as level of edema in a patient are unreliable as they are highly subjective as they are in the eye of the beholder. Researchers must ensure that these reliability errors are minimized so that if differences are seen in the data, they can be attributed to the intervention and not to sloppy weight measurements. It is with these concepts in mind that methods section in protocols and journal articles provide such extensive details related to how a study was designed and conducted. Threats to a study's validity and reliability exist at almost every turn in the research process. No one researcher can see all the potential problems, so the team approach to the discussion of validity and reliability during the development of the study design and creating and following study protocols can minimize the threats to validity and reliability. What should be the characteristic of a good measurement? An intuitive answer to this question is that the tool should be an accurate indicator of what we are interested in measuring. In addition, it should be easy and efficient to use. There are three major criteria for evaluating a measurement tool: validity, reliability, and sensitivity. Validity. What is validity? Very simply, validity is the extent to which a test measures what it is supposed to measure. Question of validity is raised in the context of 3 points made above: the form of the test, the purpose of the test, and the population for whom it is intended. Therefore, we cannot ask the general question, is this a valid test? The question to ask is, how valid is this test for the decision that I need to make? Or how valid is the interpretation I propose for the test? We can divide the types of validity into logical and empirical validity. Content validity. When we want to find out if the entire content of the behavior or construct or area is represented in the test, we compare the test task with the content of the behavior. This is a logical method, not an empirical one. Example, if we want to test knowledge on American geography, it is not fair to have most questions limited to the geography of New England. Face validity. Basically, face validity refers to the degree to which a test appears to measure what it purports to measure. Criterion oriented or predictive validity. When you are expecting a future performance based on the scores obtained currently by the measure, correlate the scores obtained with the performance. The latter performance is called the criterion and the current score is the prediction. This is an empirical check on the value of the test, a criterion oriented or predictive validation. Concurrent validity. Concurrent validity is the degree to which the scores on a test are related to the scores on another already established test administered at the same time or to some other valid criterion available at the same time. Example: a new simple test is to be used in place of an old cumbersome one which is considered useful. Measurements are obtained on both at the same time. Logically, predictive and concurrent validation are the same. The term concurrent validation is used to indicate that no time lapsed between measures. Construct validity. Construct validity is the degree to which a test measures an intended hypothetical construct. Many times psychologists assess or measure abstract attributes or constructs. The process of validating the interpretation about that construct as indicated by the test score is construct validation. This can be done experimentally. Example: if we want to validate a measure of anxiety, we have a hypothesis that anxiety increases when subjects are under the threat of an electric shock. Then the threat of an electric shock should increase anxiety scores. Note, not all construct validation is this dramatic. A correlation coefficient is a statistical summary of the relation between two variables. It is the most common way of reporting the answer to such questions as the following: does this test predict performance on the job? Do these two tests measure the same thing? Do the ranks of these people today agree with their ranks a year ago? Rank correlation and product moment correlation relate to this. According to Cronbach, to the question what is a good validity coefficient, the only sensible answer is the best you can get, and it is unusual for a validity coefficient to rise above 0.6, though that is far from perfect prediction. All in all, we need to always keep in mind the contextual questions. What is the test going to be used for? How expensive is it in terms of time, energy, and money? What implications are we intending to draw from test scores? Next, we come to reliability. What is reliability? Reliability asks the question, does the instrument consistently give the same results with the same group of people under the same condition? Other words that are used synonymously with reliability are accuracy, dependability, consistency, and stability. The reliability of a measure indicates the extent to which it is without bias, that is, error free, and hence ensures consistent measurement across various items in the instrument. In other words, the reliability of a measure is an indication of the stability and consistency with which the instrument measures the concept and helps to assess the goodness of measure. Stability of measures. The ability of the measure to remain the same over time despite uncontrollable testing conditions or the state of the respondent is indicative of its stability and low vulnerability to change in the situation. This attests to its goodness because the concept is stably measured no matter when it is done. Two tests of stability are test-retest reliability and parallel form reliability. Test-retest reliability. Test-retest method of determining reliability involves administering the same scale to the same respondents at two separate times to test for stability. If the measure is stable over time, the test administered under the same conditions each time should obtain similar results. For example, suppose a researcher measures job satisfaction and finds that 64% of the population is satisfied with their jobs. If the population is satisfied with their jobs, the study is repeated a week later under similar conditions, and the researcher again finds that 64% of the population is satisfied with their jobs. It appears that the measure has repeatability. The high stability correlation or consistency between the two measures at time 1 and at time 2 indicate high degree of reliability. This was at the aggregate level. The same exercise can be applied at the individual level. When the measuring instrument produces unpredictable results from one testing to the next, the results are said to be unreliable because of error in measurement. Parallel form reliability. When responses on two comparable sets of measures tapping the same construct are highly correlated, we have parallel form reliability. It is also called equivalent form reliability. Both forms have similar items and same response format, the only changes being the wording and the order or sequence of the questions. What we try to establish here is error variability resulting from wording and ordering of questions. If two such comparable forms are highly correlated, we may be fairly certain that the measures are reasonably reliable with minimal error variance caused by wording, ordering, or other factors. Internal consistency of measures. Internal consistency of measures is indicative of the homogeneity of the items in the measure that tap the construct. In other words, the items should hang together as a set and be capable of independently measuring the same concept so that the respondents attach the same overall meaning to each of the items. This can be seen by examining if the items and the subsets of the items in the measuring instrument are highly correlated. Consistency can be examined through the inter-item consistency reliability and split half reliability. Inter-item consistency reliability. This is a test of consistency of respondents' answers to all the items in a measure to the degree that items are independent measures of the same concept. They will be correlated with one another. Split half reliability. Split half reliability reflects the correlations between two halves of an instrument. The estimates could vary depending on how the items in the measure are split into two halves. The technique of splitting halves is the most basic method for checking internal consistency when measures contain a large number of items. In the split half method, the researcher may take the results obtained from one half of the scale items, example odd numbered items, and check them against the results for the other half of the items, example the even numbered items. The high correlation tells us there is similarity or homogeneity among its items. Sensitivity. The sensitivity of a scale is an important measurement concept, particularly when change in attitude or other hypothetical constructs are under investigation. Sensitivity refers to an instrument's ability to accurately measure variability in stimuli or responses. A dichotomous response category such as agree or disagree does not allow the recording of subtle attitude changes. A more sensitive measure with numerous items on the scale may be needed. For example, adding strongly agree, mildly agree, neither agree nor disagree, mildly disagree, and strongly disagree as categories increase a scale's sensitivity. The sensitivity of a scale based on a single question or single item can also be increased by adding additional questions or items. In other words, because index measures allow for greater range of possible scores, they are more sensitive than single items. Practicality. The scientific requirements of a project call for the measurement process to be reliable and valid, while the operational requirements call for it to be practical. Practicality has been defined as economy, convenience, and interpretability. Establishing the validity of the instrument. We shall now see how you can establish the validity of your instrument. Establishing the validity of an instrument includes a review of the instrument by a panel of experts and a field test. The general procedures are as follows. Clearly define what you want to measure. Dollars spent, knowledge level, attitude, people involvement, behavior change, and so on. Locate or develop questions to include in the instrument. Search for existing instruments related to your evaluation, focus, and topic of interest. These may be useful as a guide in developing the new instrument. Develop a draft of the instrument in consultation with some of your primary stakeholders and colleagues. Select 5 to 7 people to serve as a panel of experts. These people should be knowledgeable in the specific content area of the instrument. This group will review the instrument in terms of content, format, and audience appropriateness. It is very important that you inform the panel of the purpose of the instrument. Once the panel reviews the instrument and provides you with comments and suggestions for revision, revise the instrument using the panel's comments for guidance. Conduct a field test to establish the validity of the instrument in terms of suitability, utility, and clarity. Select 8 to 10 people to participate in a field test to review the instrument for clarity, content, wording, and length. These people should have characteristics similar to those of the target audience. For example, if you plan to have farmers as your target population, then the field test, the questionnaire with a different group of farmers in an adjacent watershed. Once the field test results are received, address these comments as you revise the instrument. After the validity is established, the reliability of the instrument needs to be tested through a pilot group. The people in the pilot group should not be the same participants from your field testing. Next, we come to establishing the reliability of the instrument. How do you establish the reliability of the instrument? Pilot tests are used to establish reliability of the instrument in terms of accuracy and consistency. The general procedures are as follows. Compare the scores referred to as paired scores for each question on the two sets of instruments. A high percentage of agreement between the paired scores indicates a reliable instrument. Use 70% as an acceptable percentage level. Questions within the instrument receiving low percentage agreement need to be revised or deleted. If major revision is required, the instrument's validity testing will need to be repeated. Select 15 to 20 people to participate in the pilot test. They will be asked to complete the entire instrument twice. These people should have characteristics similar to those of your target population. Administer the instrument to this group. Within 7 to 10 days, administer the same instrument to the same group. To conclude, I am sure you have understood that research data collecting instruments must be validated and checked for reliability. This will ensure that your effort to do research would be commendable. Otherwise, it raises queries by those evaluating your work and those using it to design their own work. If you are using measuring devices like, for example, weighing scale, then it requires calibration. The same is true for laboratory instruments. It is always advisable to run a standard or a solution of known concentration along with the test sample.